So first of all, thank to the organizer. It's my third time to this kind of conference, so I always clearly enjoy, otherwise I would not be here. Always a very great pleasure. So what I'm going to discuss today is talk about trying to model epithelial tissue with a little bit higher spatial resolution than before. And okay. So what is the motivation? The motivation is that tissue, are solid, but they do exhibit some transition from a solid phase to kind of flowing one. In examples are morphogenesis or wound healing or tissue invasion and so on and so forth. So as a natural question, at least for our community, is can we describe tissue using tools and methods that are standard in statistical physics? So can we describe them in terms of a glass transition or a Jamie transition or these kind of approaches? And of course, we would like to understand what are the control parameters, what is controlling this, their behavior. And also, we know that tissue are not standard thermal systems, so we would like to understand at the microscopic scale what is unique, what is actually making tissue a living system. And there are different approaches to this question. So in the biological community, what has been done, and this has brought a lot of insight, is the so-called top-down approach. I will call it learning by breaking. So to understand how a biological system works, what you usually do, you go there and you perturb. You suppress the expression of a gene, and you see what is the consequence on a microscopic individual to understand what is the role of the gene. Another approach, which is more physically inspired, is the top-down approach, in which you try to build models. And this has become extremely popular in the community in, perhaps I can do from here, in the last decades, mainly because we had very important advancement from the experimental viewpoint. So we are now able to image tissue with a very high spatial resolution. And so we know where cells are, and also inside the cell, the dynamics of the cytoskeleton. We have more recent insight about the mechanics. It's now possible to measure, at least to have insight on what are the forces between the forces between cells. So for instance, now we know that tissue most often are under a state of tensile stresses. And then from the theoretical viewpoint, we had a lot of insight from non-equilibrium physics, in particular from the branch of active matter. So if you want to try to make a model of tissue, you have to think of what are the basic ingredients, and the basic ingredient is, of course, the cell. What is a cell? A cell is, from the physical viewpoint, is a colloidal scale particle, which, however, is not rigid, is actually deformable. So you need to think about, okay, is the deformability important? If so, how many degrees of freedom I need to use to describe this deformability, and how I use them practically. And then you want to discuss the dynamics, and you know that the way cells move is through the dynamics driven by the lamellipodium. That is, cells have not only a bottom-up symmetry, but a front-back symmetry. And the way they work, they extend at the front part, they extend some protrusion, the protrusion grabs to the substrate, and then the cell pulls itself forward, and so on and so forth. So this is lamellipodium. So if this is important, if so, you have to think about how to describe. And then, of course, you have a tissue which is a collection of cells. So you have many cells, and the cells are interacting. So you need to describe this interaction, which are mediated by the cell-cell junction. So the typical length of this cell-cell junction is much smaller than the typical size of the cell. So these are kind of sticky particles, if you want. And so you have to describe this kind of interaction. And another thing you have to do is to describe something that I'm going to focus a lot in this talk, which is different kind of sensing. So how a cell is able to sense the environment and to respond to it. So of course, there have been a lot of models that are trying to mimic this kind of behavior. And here I'm just listing a few of them, like the POTS model, where you put cell on, each cell is a collection of sites of some color. So here is the figure just represented one cell. And you can think about some kind of dynamics, which is a very simple model, but of course it's has its limitation when you study the dynamics. So what is time, or you study the elasticity, is very difficult. Or you can have a center-based model, which each cell is kind of Lennard-Jones particle. So you are totally missing the deformability of the particle. So is it important or no? How do you cope with it? On the other side, you have a model like the Voronoi and the vertex model. We have heard about it yesterday. Well, these are not models for cell. These are models for tessellation, meaning that you only have a cell because you have a neighbor. Okay, so you cannot model an isolated cell. If you want to do that, you have to develop a very complicated model. And then I have to say this model have kind of behavior that is difficult to rationalize. For instance, you start compressing this model and they fluidize. So say, okay, perhaps there is something that is not really physical. Or there is a recent addition to the zoology is the phase field model, where you describe its cell as a concentration gradient. This is a very nice model, but computationally they are very, very expensive. So you can simulate just a very small number of cells. And there have been, of course, many efforts to try to mix model or to try to extend this to try to mitigate the limitation. And one consequence of this, somehow, this plethora of models is that there is actually not an agreement on what control very simple basic feature of the behavioral tissue. So, for instance, you may want to ask what is actually causing motility and what is actually trying to slow down motion. So there is one thing in which everyone agree is that the motility is promoting tissue motion. You ramp up the motility and cells tend to move. But then you may think, okay, what is the effect of crowding? So intuitively you may think, okay, I compress the tissue and the tissue the dynamic is slowing down. And you see this in some experiment. This is reproduced by particle-based model, but you also have experiment that are suggesting the converse, and you have models, the Voronoi and the vertex model, that is suggesting that you compress the tissue and the dynamics might speed up, which is a little bit weird. Or you may think, what is the effect of adhesion? And again, you find experiment and some model that suggests what you will say. The first thing you may think of is that if adhesion increases, then dynamics slow down, particles stick to each other. But it is also found or suggested the converse. And about sensing, we're going to focus on a particular model of sensing, which is called contact inhibition locomotion. So the name suggests you that upon contact, the cells should stop moving. This is suggested by some experiment, by the first experiment that have been introduced in this sensing mechanism. But then you also have experiment that suggests the converse. And when people try to encode this sensing mechanism in models, they find that it's actually promoting tissue motion. Okay, so there is, on very basic things, there is not an agreement. Let me say that from the model viewpoint, things are complicated, but from the experimental viewpoint, are even more complicated. And the reason is that we like this kind of schematic description, like adhesion, density, or sensing, but experimentally, these are not control parameters that you can tune independently. Okay, so for instance, if you want to tune adhesion, what you usually do, you add some drag that is interacting with the expression of cadherin, molecules in the cadherin family, which are involved in the formation of junction. But the same molecules are involved in the sensing, in contact inhibition. So you perturb adhesion, but you also perturb sensing. So you don't know precisely what is happening. Okay, so what we are going to do here is to try to develop a new model with, I'd say, a little bit of subcellular resolution. And then, since I want to understand what is driving a tissue out of equilibrium, the first thing we do is to have a reference. So we study the equilibrium phase behavior, and then we move out of equilibrium and we study what is the effect of motility, densing, adhesion, and contact inhibition. And then we're going to focus a little bit on wound healing, and we are going to suggest that it's possible to introduce the concept of force chains also in this kind of soft, squishy particles. So the model that we introduce, we call it Next Tissue. We call it Next Tissue because NTU is the acronym of my home institution, Nanyang Technological University, so I have some edge when I beg for money. And it is an extension of LAMPS, so which is a very popular package, and if you are interested, you can just take and download it. And the starting point is related to what we have seen in the last talk yesterday. So it's a model introduced by the group of Corio, which is the deformable particle model. So we are in 2 dimensions. And so each particle is described as a ring polymer. So in the energy functional, you have a term here, which is just the sum over all the spring. But then you don't want the polymer to collapse. And then you add another term, which is a penalty related to the dependence of the area with respect to some preferred value H0, okay? And you can non-dimensionalize this energy function for one single particle, and it turns out that it depends on, very similar to the Voronoi vertex models, depends on one parameter. You can introduce P0, which is the ratio between essentially the preferred value of the perimeter, which is the number of bits, and L0 is the typical distance, over the square root of area. And we're going to focus here on a value which is somehow large, 3.9, means that this particle are very squishy. So their individual shear modulus is very, very small. Let me say that the model resemble a lot. We can change as long as we are soft above some threshold. That's not really, it's not really important. One thing that we like to highlight is that this P0 in this model is affecting the properties of a single cell. This is very different from what you have in the vertex and body model, where the P0 combine the property of the cell and the property of the interaction all together in one parameter. Here we have two different things. P0 is property of the cell. How about the interaction? The interaction of the cell, as we mentioned, is driven by the formation of adhesion molecules. And so what we say that between the membranes bits of two cells, we are interacting, adding an interaction potential, which is an attractive potential. So overall, the interaction potential between monomers of different particles is going to be 0 if they're far away, then you have some attraction, and then of course you have repulsion because you don't want interpenetration, okay? The key point here I would like to highlight is that this attraction range is very small compared to the size of the diameter. So the blue is very small compared to the particle size. So we are in the regime of sticky particle. The particle only interact when you're very close, okay? Already with this model, yes, Silke. Yes, we do have problem in which the surface here might buckle. This might happen, but you have to go to very high density. So if the volume fractions don't go to very high, we are okay. We can fix, in principle, if one essentially following what has been discussed yesterday, so you can add some kind of angular spring to prevent this thing. But we are not going to this by high value the density, so we are not going to, we already have too many parameters, so we try to not to add other things. Okay, so already with this model, you can study very simple things. So this is a very famous experiment by Chara's group, in which they grow a tissue on a plate, then they detach it, so you have a suspended monolayer, and then they pull and stretch. So here there is no dynamics, essentially, if you want, it's a quasi-static induced fracture. And in the model, you see possible animation, you can do the same thing. You prepare your tissue, and then you stretch it, and you see you have a confluence tissue that at some point it fractured. Or this one? Yeah. So one special feature that was is mentioned in the experiment is that if you look at what is the strain, the elongation at which the system breaks, is large. It's very large. So it's the order of 1 or 0.6 or something like this, which is huge compared to the one of molecular solid or colloid solid. This
gels.
It might similar to be the one of gels. Yeah. Related to the formability.
As a bunch of sort of.
No, possibly you don't see here. Possibly here there is. So the color cells are in blue when they are interacting. So here possibly you don't see there are the gray cells because these are the free boundary. Okay. So they're here and they are on the left, on the top. I mean the free boundary. Possibly you don't see them, but okay. Or you can do the shearing. This is related to what was done yesterday. So we start with essentially what was a crystal, and then you shear. You're essentially always confluent, and you see the deformation of the cell, and you're going to see clear emergence of nematic ordering and these kind of things. Okay, but in the talk here, we're not going to focus on the mechanics. We're going to focus most on the phase behavior. So the first thing I want to do is to have a reference.
Is that something that's seen in tissue, or it's?
It is something that is seen in model tissue. Yes, there is a lot of discussion about the nematic order induced by deformation. Yeah. Well, there are not actually experiments that are actually doing this kind of protocols. This is more our style than the biological style. Okay, so the equilibrium phase diagram. So what we're going to do is a very standard thing. So we have a system of 1,000 cells, each of them with 50 monomers. And we apply a stochastic force to the center of mass, and then essentially we do a Langevin dynamics. We ensure that we are in equilibrium, so long enough simulation, and then we characterize the phase. And what do you find for the phase diagram is these things here. So here we're plotting on this axis the temperature in units of the strength of the attraction between the monomers. So if you are a high temperature and you increase the density, what you see that the dynamics is slowing down. The color code is the diffusion coefficient. And then at some point you enter a glassy regime. You can define whatever you want. Or if you fix the density and you cool down the temperature, you enter in a regime of phase separation. And this is just, for instance, an illustration of what you see in the coexistence line. So one thing that you notice is that you have the coexistence between a very dense phase and essentially an empty gas, right? Which is what you see here. You have the coexistence between essentially an empty liquid and a dense one. Okay, and here let me say that on this axis we have the density, but I'm actually plotting the density in terms of the so-called effective volume fraction, which is the volume fraction the system would have assuming that the size of the particle does not change, remains the one in a dilute condition, okay? If you think about the actual volume fraction is bounded by 1. You cannot really go above 1, but this one can go above 1, and 1 is essentially is what you expect at confluence, okay? Okay, so another thing I'd like to notice is that this phase diagram is essentially very, very similar to the phase diagram that people observe in a system of hard-colloidal particle with sticky interaction, okay? There are some minor differences I'm going to discuss, but it's very, very similar. So the main message is that the softness is not really inducing a new physics, at least from this point of view. There are some differences, but...
So Massimo, one question.
Where are you? Oh, yeah, sorry.
D is diffusivity, that particle diffusivity. What is D?
D is the particle diffusivity.
Diffusivity.
This is, yes. I'm going to comment then on this. Okay, so now I would like to drive the system out of equilibrium to see how things are going to change for the phase diagram. And the first thing that we're going to do, we are going to introduce some motility. Okay, so no temperature, but motility. How do we do that? In the following, we attach to each cell what is the center of mass, and to the center of mass we attach a polarity, n, and then given the polarity and the position of each monomer with respect to the center of mass, we can define an angle phi. And this phi allows us to identify the monomers that are in front of the cell, which here are some other one in orange, from the monomers that are on the back. So the way we model this lamelliponium-induced dynamics is to say, well, the active forces are only acting on the monomers that are in front. And we also assume here that the active forces are parallel to R. Another option would be parallel to N, but parallel to R appears more reasonable to us. And there are differences in the typical shape that you get, these kind of things. And then the cell move, and there is, of course, a viscous damping due to the interaction with the substrate. So in terms of parameter, one thing to realize is that when you study healthy cell and you focus on what is the typical velocity and rotational diffusion coefficient for the motion of the polarity, what this gives rise to a persistent length that turns out to be of the order of 1 cell diameter. Okay, so Peclet number is very, very small. For healthy cell. You can add drugs to change this, but for healthy cell, this is the number you get. So if you study, we're going to study the phase diagram. On one axis, we're going to have the density, and the other one, we have this motile force that we're going to normalize. We're going to normalize with the minimum force you need to apply to detach two molecules, two cells, okay? And this is useful because there are experiments that are studying this and tell us that the typical Hilting force between two cells is of the order of 10 to the minus 7 Newton. So we have a kind of way to relate to experiment. And this is the comparison between the two phase diagram. This is the thermal phase diagram, and this is the phase diagram just induced by this motility. And what you see is that they are very similar, right? So this is the main message. And why they are very similar, and the reason is very, very simple, because yes, we are out of equilibrium, but the Peclet number is very, very small. The persistence length is 1, so we don't expect, for instance, we are way below the typical persistent length at which you will have motility-induced phase separation in 2 dimensions, okay? So you don't expect important changes, okay? So motility is there, but it's not really key. Okay, so now we're going to discuss sensing, and sensing turns out to be a very important aspect of living matter. So the molecular biology of the cell, which is kind of the only text in the community, state that the essence of life lies in its ability to sense the environment, process information, and generate appropriate response. And of course, it's very well known that cells have some sensing ability. One thing that is very well established is that cells are able to respond to gradients, like chemical gradients, or also gradients in the stiffness of the substrate. They like to go towards the stiffer region. Here, we're going to focus on another kind of sensing, so we don't have inhomogeneities in the space, but we're going to study the sensing of the interaction between the molecules. So how a molecule responds, our cell respond to the interaction to another. And we're going to focus in particular on so-called contact inhibition of locomotion. So let me say that many sensing mechanisms have been proposed in the community, but then when you dig down and you ask what is the sensing mechanism that has been experimentally proven, in the sense that it's known precisely what is the biological pathways that is activating during sensing and then is causing a mechanical response. Well, this is essentially the only one. There is contact inhibition, then contact of reproduction, inhibition of reproduction. And so this is the reason why we only introduced this one in the model. So how does it work here? Consider the interaction, the collision between 2 cells. Here are the lamellipodia at the front. So the cell established contact. So here they form junction. They adhere to each other. In response to this, a cascade of biological pathways that is activated inside the cell. And the response that you get is that the extension of the protrusion in the direction of contact is inhibited, while the extension in other direction is still there. And so the consequence is that the cell is going to rotate, okay? So this allows you to resolve the contact. And contact inhibition has been shown to slow down the motion in the suspension of sick fibroblasts. This is what Abercrombie introduced. And Abercrombie suggested that it's actually involved in cancer invasion, because essentially, since it's inhibiting motion, he said, okay, if you have a dysfunctional seal, then the cell can actually are able to invade. They don't stop their motion. On the other side, since this is a way that cells have to resolve their contacts, it is also expected that it's very useful to promote tissue invasion, because the cell, instead of trying to push towards the neighbor, are going to move toward the free space, okay? Because they want to resolve their contact. They orient toward the free space. So the way we model is to essentially to a non-reciprocal interaction between cells. So consider again the interaction between 2 cells. Only the lamellipodium is sensing, only the front part. So for instance, in our case, the top cell has a free lamellipodium, so it's not affected by this interaction in terms of the polarity. How about the cell that is below? The cell that is below does not want to extend its lamellipodium in this direction, in the direction of contact, but is still able to extend in the other direction. What is the other direction? This is the direction of the free space probed by the lamellipodium, okay? So it's not extending the lamellipodium on the back, but it's expecting in the direction of free space probed by the lamellipodium. So what we do, we identify this direction, and then we introduce an alignment interaction between the polarity and this direction, which we define somehow as the direction of the free space probed by the lamellipodium. So when you do that, you need to introduce a typical time scale for the alignment, and also an alignment strength. And the alignment strength is going to depend on the structure, because when you don't have any free space, you are not going to have any alignment, okay? So this beta is going to depend on the structure, how many monomers are in contact, these kind of things, and we have an answer for this. Yes?
Just a clarification. Is the P what was the N hat earlier? Is the polarization what?
This beta here.
P.
A p depolarization.
Yeah, is that n hat?
Yeah.
So it's the same polarization, right?
Yes. There is another question on the back.
So that looks actually quite similar to another model which has been living in literature occasionally, where you orient with the force that's being exerted on you, so force alignment. Do you think it's similar?
I don't think it's similar. When you do force alignment, you induce a lot of flocking that you don't induce here. So possibly there is another mechanism that is induced flocking. Here is not, you're not inducing it. Okay, so now we introduce this regime. We have essentially already studied the case in which this tau c is very, very large, so the alignment plays no role. Is the case we have seen before, just motility. And now we're going to go to study the other limit in which this term is dominating, and so we can compare the two regimes.
So the detection of free space, all these other detections also were possible. For example, to the opposite direction. How do you choose which direction to take?
Yeah, so we only look, we didn't identify this vector, this RC, as the free space that is probed by the molecules that are here represented in orange, and they are not in contact. So this is the free space probed by the lamellipodium. Why we do that? Because SIL, contact inhibition, is doing exactly that. The motion is induced only by the front, which for us will be the orange. Here is where you have protrusion extension, not on the back, only in the front. And during contact inhibition, only part of this protrusion extension is inhibited. Okay? So essentially we say, okay, you cannot move here, but you move in this direction. And what is the direction? Is the direction of the free space probed by the front. Okay? Yes, so it needs to turn slightly. But that slide.
Because the cells of the bulb would have no such space.
When you, yes, you are totally right. So indeed, this is something that is coming from this beta. So beta is going to depend on how many neighbors you have. Okay. So if you don't have any free space, you're not going to have any alignment, right? Because you align toward the free space, there is no free space. So when you have no free space, this beta is going to 0. Okay. So when you study the phase diagram, what you find? Well, you start to see important differences. So again, what we found? We found that while in the absence of SIL, we have a huge region of phase separation. In the presence of this contact inhibition, the region of phase separation is suppressed a lot. And so we can say that contact inhibition is promoting the homogeneous phase. So the critical point, if we want to use this terminology, is suppressed, and also the amplitude the width of the consistence decreases. Or equivalently, we can say that contact inhibition is going to suppress density fluctuation. I'm going to go back to this point. How about the dynamics? So to compare the dynamics, what I'm going to show you now is comparing how the diffusivity varies as we increase the density along this line, so in the absence of SIL, and this other line in the presence of SIL. Okay? So we can see, is SIL speeding up or slowing down the dynamics? And this is what you get. So this is the diffusivity in absence of contact inhibition, so the dynamics slowing down as you increase the density. When you switch on contact inhibition, you see that the red points are below the blue one. So the main message is that where contact inhibition is slowing down the dynamics. You see here we have a kind of weird kink, some strange behavior. So what's happening here is related to the question we had before, is that contact inhibition acts when you have free space. When you start to be very close to confluence, you no longer have free space, so this effect all of the point is going to vanish. So you expect that above confluence, the two diffusivities need to match. Okay? So this is why it's going up. So can we understand, I'm a little bit late, can we understand how to model this? And the main assumption is that, well, we can say that what's happened when you have a contact due to contact inhibition is that a cell is experiencing a contact and is turning. So contact inhibition is changing the rotational diffusivity. If you only assume that this is what is happening, so you don't assume that you have any change in the structure, then you can actually measure what is the typical decorrelation time for the polarity, and you can use this, and you say, well, the diffusivity in the presence of contact inhibition is the one you have in the absence of contact inhibition but with essentially you are changing the rotational time scale. And when you do that, you get these points here, which somehow capture the typical behavior. So the main effect of contact inhibition is change in polarization time scale here. Okay, and how about the phase diagram? Well, it's easy to understand what's happening in some aspects. So for instance, in the absence of seal, if you consider point A, you're going to see cluster. If you switch on contact inhibition, the polarity of the particle, which are represented by the color here, are not going to be random, but are going to be directed toward the free space. So if you start with a cluster, but all polarity orient toward the free space, you're going to exert tensile forces on the cluster, and the cluster might break, which is what you see here. If you have, again, in the consistent phase, but you are a high density, what you expect to observe, you have cavities. In the absence of contact inhibition, again, all the polarity are random. In the presence of contact inhibition, all the polarity points toward the free space. So again, you are trying to close the cavity, right? So you're trying to suppress density fluctuation. And so you put the tissue under tensile stress. And in this case, you're still in the equiescence region, so that these forces are not large enough to deform the cell and close the gap. But if you move to point C, that for some reason correspond to these things here, what you're going to see that in the absence of contact inhibition, you still have gap, but with contact inhibition, you close the gap. Okay? So it's promoting healing. And it's promoting healing. This means that it's changing the location of the confluence line. Contact inhibition is promoting confluence. And it's doing that by swelling the cell. Here we don't have reproduction, but you put the tissue under tensile forces, and because of this, you have a little bit of elasticity, and the cell can increase in size. And the typical change that we observe is below 7%, and this is similar to what you see in experiment when you suppress reproduction. Let me finish just mentioning wound healing and force chains. So one thing that you can do with this kind of model is to study force on the membranes, and this is, I guess, quite nice. So here, for instance, what I'm going to do is to color code the monomers according to the direction of the force of interaction of the monomer compared to the position of the monomer with respect to the center of mass. So essentially, the red means that the monomer is under tension. You're trying to locally swell the particle, where blue is under compression. And this is an animation of wound healing. So this it's a section of a very, very long channel, and there is no reproduction. So you're going to see wound healing only because the size of the particle are increasing a little bit. And you start with a condition in which you see red and blue. So essentially, this is kind of zero pressure. There are no tensile stresses. And what's happened as time goes on, the cell on the boundary are going to polarize toward the free space. They drag the others. And everything is becoming, you go to confluence, and everything is becoming red, meaning that the system is suppressing density fluctuation and is putting itself under tension. Okay? So seal is a mechanism that somehow tissue, they aid so much density fluctuation that, at least in this model, that they put themselves under tension to suppress them. This is what they are going to do, which is, of course, not standard. And this is the dynamics. Perhaps I can skip. Instead, I would like to say something about the forces. So you can also think about what is the force that one cell is exerting on the other cell. How do you do that? Well, you just sum up the force of interaction between the interacting monomers, right? And then you can think, okay, what is this force with respect to the line that is connecting the center of masses? And if you study the scalar product, you find that it's essentially it's peaked at 1 or -1, meaning that either you have forces that are parallel to the direction of separation or anti-parallel. So essentially, it is a too good approximation. You don't have frictional forces, which is similar to what has been reported by Corey. And this indicates that you can actually draw the forces as lines that are joining the center of mass, as so-called force chains, and to color code according to their strength, and the width here represents the strength, and the color represents the magnitude. And then you can study the distribution in different conditions. So here we're seeing what's happening before healing. We are in the red case. We have exponential tail, both for strong repulsive and strong attractive forces, or after healing, we still have the same phenomenology, but with larger forces. So in any case, what I want to highlight is that possibly this model might open up a more detailed description of the mechanics of tissue. So I would like to close. And essentially what we have been doing here, we have introduced a model for cell their interaction with a little bit of subcellular resolution. We model a little bit more precisely essentially the membranes. And what we have seen is that the key ingredient that gives rise to a new phenomenology in this model is contact inhibition of locomotion. It's a non-reciprocal sensing response. And what it's actually doing practically is opposing density fluctuation. You're trying to suppress density fluctuation, so this is very different from the usual motility-induced transformation. This is doing just the opposite. Perhaps I can take questions if any. Thank you so much.
So is there a unique way to model CIL, or there are many options to model CIL?
So this is what we have been doing. So when the other way people have been doing is more like a kind of alignment interaction, but then you go into flocking. So when you do that, you find that contact inhibition is promoting motion in dense tissue, which is a bit weird given the name contact inhibition or locomotion, right? So this is the only way, yeah.
It's very interesting. So a specific question. When you do you have to graph with the forces, where you define the forces between cells.
Yes. This one?
The one before? No, the other way. That one, yeah?
Yes.
So usually you also get tension along junctions, so not with cell like this, but contractile.
Yeah,
Is
yeah.
this something that's possible in your model as well?
Yes, this of course, because this is a ring polymer. So we have the spring between the bits, so you can measure we are focusing on this. This is essentially because this is novel. The tension you can measure and we can compare, but this is something that is not usually accessible in the other models. So this is why.
So I'd be interested to see, because you see often force chains along junctions instead of the way you
for
were
the- yeah,
doing earlier.
Yeah.
Have you
Something we
looked
can
at
do.
this?
No, we are not looking at this into details. Yeah, but we can do that.
So there are some report that the wound healing dynamics sometimes depends on the geometry of the wound. Did you check any of those?
No, what I can tell you is that for sure you have different mechanisms. So this is what we have is wound healing driven by motility. In particularly in the embryonal state, this might not be the case. It's most like the tensional junction, the pull string mechanism that is driving the dynamics. So you may have different mechanisms that works in different contexts. Yeah.
And another quick question. So in the only one cell scenario, does the motility direction still changes there, or is it?
It's random diffusion.
Yeah, but that is random.
Random diffusion, random rotational diffusion. The standard.
This cell motions has this different kind of taxis. I mean, typically in like wound healings, I think like this galvanotaxis and these things matters. So here, these motions.
We are not introducing any gradient of anything. So this is just interaction. Yes.
Okay, so like it's quite general. Like.
Yeah. It's quite. We don't introduce this. Yeah.